Hi, this is Mr. Anderson, and I'm sitting in my kitchen. I'll talk a little bit later in the podcast about why I'm sitting in my kitchen, but we'll get there later. Chapter 10 is about molecular biology of the gene, or more importantly, it's about this molecule, DNA. So you should have an understanding of what DNA is and how it's used in this chapter to make proteins eventually. Okay, let's go back a little bit, simple history of DNA. It all began with an experiment by Frederick Griffith way back in the day. This is around World War I, and he was trying to study bacteria, or infectional bacteria, and he was looking at these types of bacteria. We call them S and R because they're smooth or rough. Smooth bacteria would kill a mouse when injected, but rough would not. He then took this smooth bacteria, which is normally virulent. He would then heat damage it. So he'd heat it up. It would actually, we know now, denature a lot of the materials in there. He would inject it in the mouse. The mouse was good to go. And so this all makes sense at that time until he actually adds the normally non-virulent to the heat-damaged virulent bacteria. He just lets them mix together for a while, and then he finds that they kill the mouse. So that's kind of a discrepant event. So what's going on here? Somehow, he said, it's that bad, dead bacteria is transforming the good bacteria. We now know that it was actually DNA that was being passed. Fast forward a little bit after World War II, and they have a little more time to look at this problem. Hershey and Chase are studying phages, and a phage is a virus that actually injects material into a bacteria, and it allows that to make copies of itself. Hershey and Chase want to figure out what in the virus is actually being passed from generation to generation. Is it the outside? All viruses have a protein coat around the outside, or is it the genetic material on the inside? In this case, it's DNA. So they want to figure out, is it the proteins or the DNA that's actually being passed? What's the secret of life? What's this code of life? And they use an elegant experiment where they dye the outside with sulfur, because sulfur is not found in DNA, but it's found a lot in proteins. And then inside the DNA they dye that phosphorus. Phosphorus is found in DNA in just tiny amounts in protein. They then put it in a kitchen blender after it's infected the bacteria, and then they centrifuge it, and they find at the bottom of that centrifuge tube that it's actually the DNA that's being passed. So now we have this mad rush to figure out what's the structure of that. And you now know that Watson and Crick win this race. They borrow this picture from Rosalind Franklin with the help of her co-worker Maurice Wilkins, and they figure out the structure of DNA. Became the most famous biologists of their time. James Watson is still around today. And so this is the model of DNA that they came up with. What is it made up of? Well, this thing right here, the thing that I'm now drawing around, is something called a nucleotide. A nucleotide is made up of three parts. You have a sugar, in this case it's called deoxyribose sugar. You have a phosphate group, that's one of our functional groups we learned earlier. And then this, which is a nitrogenous base. Nitrogenous base comes in four different types: adenine, thymine, cytosine, and guanine. And there's this base pairing rule called Chargaff's rule that Watson and Crick actually used to figure this out. The amount of adenine and the amount of thymine are always equal because they bond to each other. Now the thing that I wanted to point out on this diagram is that this backbone and this backbone are not only parallel to each other, in other words they run right next to each other, but they are anti-parallel. In other words, they're parallel, but they run in opposite directions. And what directions do they run in? Well, if you look down here, this carbon is actually called the 3 carbon, or the 3' carbon, and this carbon up here is called the 5' carbon, so we can count it out: 1, 2, 3, 4, 5. And so this backbone is said to go from the 3' to the 5', whereas this one goes from the 5' to the 3', so they run in opposite directions. Okay. What's the first thing they suggest in their one-page paper in Nature? Well, this suggests, they say, they say something like, it has not escaped our attention that this structure suggests a form that could be copied and also could then contain a message. And so first thing we want to do is copy our DNA. And when do we do that? Remember in mitosis and meiosis that during interphase we have to copy all of the chromosomes and make sister chromatids from that. And so the first time we have to copy our DNA, it's very simple. All you do is unzip the DNA and then add a complementary strand on the other side. So to make this simple, if there's an adenine and a thymine and a thiamine, or an ATT on this side, there will be a TAA on this side. Now here's our 5' and 3' abbreviations, and so the DNA will simply unzip in the middle, and then we add complementary strands. Now the protein that does that is something called DNA polymerase, and it's an enzyme that actually adds the new letters to the DNA. On the top strand right here you can see that those letters go on silky smooth. We sometimes refer to this as the leading strand. DNA polymerase goes in this direction as the fork starts to open up. On the other side though, you can see that it's going in the opposite direction. As the fork opens up, the DNA polymerase will actually go backwards to the direction of the fork. Reason why, and this is one of the things you should always remember, we can only add the DNA nucleotides to the 3' end of the DNA. And so we can't add it right here. And so after we've opened up that fork, on the lagging strand will actually go in the opposite direction. Then another enzyme that you should remember called DNA ligase will actually come in, and it ligates the DNA, and ligate just means attaches those fragments together. Now if we were to start on one side of our genome or one side of all of our chromosomes and go to the other side, it would take forever to do that. And so instead we use what are called bubbles, and the bubbles will start all along our DNA. And right here then you can see one replication fork, which if we zoom into it would actually look like that. So you have all these sites where it starts and then they work towards each other. Okay, now I'm in my kitchen. The reason I'm in my kitchen is that DNA is nothing more than a cookbook. It has a bunch of different recipes in it, but think of it as a special cookbook that can never leave a special room. It never can even go to the kitchen. It's just locked away like a special book. So it's got all these great recipes in it, like recipes to make keratin or hemoglobin or whatever. And so that's all the recipes, and we call those genes. So how do we actually go from a gene to something that we use in the body? In other words, how do we go from a genotype to a phenotype? We use something called RNA. And now RNA is different from DNA in 3 ways. Number 1, it's a single strand. Number 2, it has a different sugar. It's called a ribose versus deoxyribose. And the third way that it's different is that we actually have a different base. Instead of thymine, we have uracil. But it serves the same purpose, and we think it was the first hereditary material on our planet. So the first process is called transcription. What you'll do is you'll find a recipe that you like, and then you simply transcribe that. And so you transcribe a recipe, or you write it down. And that transcript is called messenger RNA. So in this diagram right here, this pink molecule is actually RNA that's copying itself off of one side of that. We call that the sense strand of DNA. Now that recipe, once we have it on that little card, is able to go to the kitchen. But before it goes to the kitchen, we do a couple of things to it. First thing we do is we actually add a cap and a tail on it, and it protects it. And then we splice out these sections called introns, and we'll talk about why we do that later. But we have to do some post-transcriptional modification. So we modify the messenger RNA, but then on its way to the kitchen. And we make dozens and dozens and dozens of these, and they all go to the kitchen. Once you're in the kitchen, what do we actually do in the kitchen? Well, this is a recipe to make brownies. And so in the kitchen, we're actually going to put together all the ingredients to make something. That'd be brownies in a kitchen, but in the DNA kitchen, or the cytoplasm, or excuse me, in the cellular kitchen, we're actually going to use amino acids to spin together and make something called a protein. We have to talk about 2 more types of RNA here. This is called transfer RNA, and then this is ribosomal RNA that actually makes up the ribosome. So transverse RNA's job is to bring in a specific amino acid. The first one is always an amino acid called methionine. Its 3 letters will match up on 3 letters of the messenger RNA, and the reason it's 3 letters is so we can code all 20 amino acids. It'll latch onto that, then we'll have the ribosome assemble around that, and the whole thing shifts over. And so as the messenger RNA feeds its way through the ribosome, transfer RNA keeps bringing ingredient after ingredient, or amino acid after amino acid, and eventually we have this long protein. Once we have a protein, that protein, remember, folds into a specific shape, and it makes something like, in this case, it would make something like a brownie. And so the brownie is something, or like a protein, that we can actually use in the cell. And so that's kind of a quick analogy for how DNA works. Again, it's got the message, it's got the recipes, but they're locked in that nucleus, and the only way we can actually make things out of it is to make messenger RNA, and then through a process called translation, or in the kitchen we call that cooking, we can actually make something like a protein or a brownie that you can actually use. So that's DNA, and I hope it helps.